information. I just want to wrap this up. And some of this material is going to be a little bit repetitive from what we talked about previously, because we're going to go back to some information about cerebrospinal fluid, and we make reference similar to serum. Now I want to go back to this illustration I had right here. Serum is going to be like the plasma component here, okay? Remember, the plasma is 90% water. The cerebrospinal fluid was derived from the plasma component. The ependymal cells bathe the brain and spinal cord to provide some nutrition. I don't care for the way the authors of this new textbook, the one that you're using right now, necessarily presents that. It seems like they're trying to create a little bit of obfuscation. That is to say, you know, giving you information that's a little bit more difficult to understand here. But we talked about cerebrospinal fluid as providing a protective cushion to protect from rapid head movements, okay? Like I mentioned to you, it's bathing, it's creating some buoyancy of the brain, 97% of the. All right, how much cerebrospinal fluid was being produced?
500.
About a half a liter in a 24-hour period of time, right? 500 milliliters. There's about 150 ml there at any one time. So the CSF fluid flow here, and this is kind of a flow chart, not that you need to know what the flow chart is, but they make reference lateral, like outside, ventricles, interventricular foramina, interventricle between the ventricles here, third ventricle, cerebral aqueducts, fourth ventricle, aperture, subarachnoid space, or arachnoid granulation, neural sinus. Fine. Nice to know. All right, moving on here, we say production. Are we good with where this is produced? I beat that into the ground earlier. Where's it coming from? Choroid plexus, right? In the ventricles, the roof of the ventricles here. Produces CSF, which fills the ventricles and other parts surrounding the brain and the spinal cord, okay? Composed of ependymal cells, their support tissue and associated vessels. The choroid plexus is those ependymal cells. What was another large title for the ependymal cells? They're glial cells, right? The ependymal cells are glial cells. They're one of those 4 glial cells of the central nerve system. Okay, and associated vessels formed by invaginations. The vascular, the pia mater, the invagination just means indentations in there. The pia mater, that's where we're going to find that. Found in each ventricle, most of the CSF is produced within the lateral ventricles. Formed by the transport of water and solutes, that should be from the blood, by following a concentration gradient. The ependymal cells of the choroid plexus actively transport sodium into the ventricles, and the water follows. This is the first introduction we have to where we talk about the obligatory pathway of water and salt. Here's what you need to know. I would pack this away for just future usage, and you need to know it if I haven't told you before. Okay, know that. That's the obligatory pathway of water and salt. The obligatory pathway of water and salt. What does it say? Where the salt goes, the water is going to follow. Okay? Where the salt goes, the water follows. You dump the salt, what are you going to dump? Water. One of the classic ways in which you should remember this is associated with people who have high blood pressure. What's one of the main things that they constantly harp on people who have high blood pressure? Reduce your what?
Sodium.
Reduce your sodium intake, reduce your salt intake. Why might that be the case? Because if you lower the salt, what should also go down? The water content, okay? So as you're going to eat salty foods, if you eat a whole lot of salty foods, what are you going to crave then?
Water.
You're going to crave water. And what will that do? All right, let's go back to that word hydrologic. Your body is a hydrologic entity here. More water, higher pressure. Less water, lower pressure. Did you follow what I just said? You increase the amount of fluid in the system, what happens to the blood pressure? It go up. What happens when you reduce the amount of fluid in the system? What should happen to the blood pressure? Should go down. So if there's one ion that they get real concerned about, it's salt, because we know that there's obligatory pathway of water and salt. Where the salt goes, what happens?
Water
The
follows.
water's going to follow. So if you increase the salt intake, you're probably gonna increase your water intake, and what'll happen to blood pressure? It has a tendency to go up. If you drop the salt concentration in the system, you go on the low salt diet, you have the tendency to have less water accumulation. Less water accumulation, lower blood pressure. Remember I told you about vasodilation, vasoconstriction? Does anybody remember that? Somebody repeated to me what I told you about that? Apparently not.
Small changes in blood vessels, big changes in blood pressure.
You gotta have diameter in there someplace.
Blood vessel diameter.
Yeah, small changes in blood vessel diameter, large changes in blood pressure. Okay, lumen size, small changes in lumen size, large changes, that is to say, you increase or decrease the size of the diameter of the vessel or the lumen size, you're gonna impact blood pressure. You increase the amount of salt, you increase the amount of water, you have the tendency to impact blood pressure, okay? Where the salt goes, the water follows. The ependymal cells of the cord plexus actually transport sodium into the ventricles. What happens? Water comes along with it, okay? Large molecules are transported by exocytosis, all right? Glucose. You heard about glucose before? You should immediately know glucose is a carbohydrate, right? Immediately make that link. What's the dominant food for fuel for the body? Come on, guys, we're too deep into the semester here for you to sit there and just say, what do you say? Glucose is a carbohydrate. What's the dominant food for fuel for the body? Carbohydrates. What's one of the dominant fuels we use for aerobic cellular respiration? Glucose. What is glucose? A carbohydrate. Are we okay? Does that make sense? You need to be able to regurgitate these things kind of at split-second notice here. Glucose, the primary energy source for the brain, mediated by GLUT1 transporters. Nice. Big deal, okay. It is a big deal. I just don't, you don't have to have that as a necessary thing for testing purposes. Here's another artist's interpretation of where that choric plexus is located in the ependymal cells part of the choric plexus, where we're going to produce the cerebrospinal fluid. All right, blood supply to the brain. The brain requires a tremendous amount of blood. Your brain, our brain, receives 15 to 20% of the blood that's pumped by the heart. That's like half, isn't it? New CLC math. What is challenging me on that? 15 to 20% is what?
A fifth. Up to a fifth.
1/5. Okay, 20%, 20, 40, 60, 80, 1/5. 1 out of 5 parts. So about 1/5 of the supply of blood coming from the heart goes directly up to fuel or supply the brain with circulation there. That's pretty impressive, or kind of important when you consider it. Interruption can cause unconsciousness and irreversible brain damage. We were just talking about that with regard to TIAs. Remember the ischemic situation? Okay, high metabolic rate. The brain has a high metabolic rate. It is constantly needing to use that fuel that's supplied to it. What did we just say the fuel for the brain was here? Glucose. The primary energy source for the brain is glucose. Glucose is a carbohydrate. Serum glucose is what we talk about with regard to blood sugar. When you have people who are diabetic and they're constantly checking their blood glucose level, they're checking the sugar concentration within the blood. Receives blood through the arteries, the internal carotid arteries. Arteries are bringing what kind of blood into that?
Oxygenated.
Oxygenated blood. Come on, guys, work with me. They're bringing oxygenated blood in, okay? Basilar arteries. Carotids join the basilar to form the cerebral arteries here. Arterial circle, and we usually refer to it goes by their name, the circle of Willis. Cerebral arteries arise from the arterial circle here. Where would you look for a vertebral artery? Remember those transverse foramina? in the cervical spinal vertebral segments. Okay, those vertebral arteries are going up through those foramen on the transverse foramen. They're passing right through there. Need to show you that when we get to the lab next time. The blood-brain barrier, we talked about that. Lining all, have tight junctions, astrocytes, we discussed that already. Mentioned there's a little bit of duplication of this information, a repeat of what we talked about earlier. Foot processes that influence capillary permeability, basement membranes. These three characteristics determine the permeability. What is permeability? Let's have it. What is it?
Some things in, some things out.
Not everything in, not everything out, okay? Permeability. Lipid soluble substances. Lipid soluble. Those are what?
Fat soluble.
Can dissolve in fats. Lipid soluble, they can dissolve in the fats. What do we have here? Pass through by diffusion. What is diffusion?
High to low concentration.
High to low concentration. Requires ATP, correct? Requires ATP? No, it's going down the concentration gradient. Notice these. Nick, what is ethanol? OL. If it's got an OL on the end of it, it's an alcohol. Ethyl is the one you go out to party with on Friday and Saturday nights. Take ethyl out on Friday and Saturday nights, okay? What's that one? That's the stuff in your brew. Okay, ethyl. Ethanol is just the, like I said, this text authors here want to elevate the vocabulary a bit. We're not going to call it alcohol, we're going to call it ethanol. Okay, ethanol. Ethanol is consumable alcohol. The safe drinking alcohol that still destroys your brain cells, okay? What's heroin? Isn't it a narcotic substance? But notice, it crosses the blood-brain barrier. Well, yeah, that's gonna give you the escalated high that they look for with regard to shooting that stuff up. Inject it into the bloodstream, crosses the blood-brain barrier, bam. Water-soluble substances move through by mediated, we talked about it before, facilitated transport, remember? Facilitated diffusion. Here they say mediated transport, same thing. Facilitated diffusion and mediated transport, just another way of saying you need to have a protein carrier molecule to move. What are we gonna move? Amino acids. Not proteins, not peptides, we're moving amino acids. We're also gonna move some glucose. What's glucose? Fuel, okay? Cranial nerves are indicated by the Roman numerals, may have one or more three functions: sensory, special or general, somatic, control of skeletal muscles, parasympathetic, regulation of the glands, smooth muscles, and cardiac muscles. When we look at the undersurface of the brain, when we take those sheep brains on Monday when we come here, we're not gonna be able to see all those. We're gonna see a few of these connections. We're gonna see the olfactory, optic, we might see a few of these other pieces in here, but we're certainly not gonna be able to count 12 different rootlets for those cranial nerves that we're gonna see there. We'll see them on the models, but we're not gonna be held accountable for these. The rest of this information, I would encourage you to go over it. It's all about those cranial nerves, and the cranial nerves are, you know, gonna be hitting you hard and smacking you around in the final exam as well. So I'd encourage you to know the cranial nerves. Homeostatic imbalances, names of them, key features about what we talked about. Here's the one, what's this one? Vestibular cochlear. Cochlear portion, hearing portion, balance portion. These are really good illustrations in that PowerPoint set. We'll deal with reflexes in the information we're gonna cover in lab 2 weeks from now. Okay? All right, I got your tests here. Get those. The average score here, oddly enough, there was 90 questions, the average was a 60.